DEG analysis produced upregulated and downregulated gene lists for each ESRRRA, including common and overlapping lists. Using the Enrichment Analysis tool in the Advanced plugin, we will first look for Go terms that may be related to the common list of upregulated genes. In the Go_term column, Go terms are separated by 3 slashes. Use the same delimiter in Enrichment Analysis. In the result table, insulin-like growth factor tilde appears at the top. Click Overlaps and press Preview to see the matching genes. This is one of the advantages of using an integrated plugin rather than a web tool. If there are other Go terms you are interested in, try pressing the Preview button to check them. Next, let's perform Go analysis on the list of genes down-regulated by 2 or more ESRRRA treatments. As we saw in the previous video, several Go terms related to the cell nucleus are found. Here again, insulin-like growth factor tilde appears. Since complex pathways often include both up and down regulated genes, this suggests a link with ESRRRA. By pressing the Save a Text File button, you can save this table as a text file. Once again, we run the analysis using the gene list upregulated by all 3 ESRRRA treatments, and export the result as a text file in the same way. We send the 2 text files to ChatGPT and ask it to extract Go terms detected in both the upregulated and downregulated gene lists. As a result, the related Go terms insulin-like growth factor tilde and insulin receptor tilde accounted for 2 of the 3 common terms. Next, we import the KEGG Insulin Signaling Pathway into Subio Platform and visualize it. The upregulated genes are concentrated in the upper half of this pathway. One gene is also found at the far left of the lower line. On the other hand, the down-regulated genes are concentrated along the lower line. This observation also suggests that this pathway is strongly related to ESRRRA. Enrichment analysis is not limited to common Go terms or pathways. The gene sets provided by GSEAA include sets grouped by various biological meanings, and these can also be used for enrichment analysis. In GSEAA's MSigDB, various gene sets are organized by category and can be downloaded for use. First, we download the KEGG_legacy subset. You can choose either the gene symbols version or the NCBI gene IDs version, but the NCBI gene IDs version tends to cause fewer problems. However, when importing this version, make sure to select Gene ID in the Select Symbol column setting of the platform list in Subio Platform. In the same way, we also download the NCBI gene IDs versions of microRNA targets and transcription factor targets. Return to the Import MSigDB Gene Sets tool window and press the Browse button. When you select GMT as the file type, the downloaded files will appear, so select and import them one by one. The imported gene sets are saved as measurement lists in Subio Platform. To keep them organized, we save them inside the MSigDB folder. Import the remaining 2 GMT files in the same way. Gene sets imported as measurement lists can be used in the Enrichment Analysis tool. Switch from the Annotation tab to the Measurement List or Pathway tab. First, set the gene list upregulated by all 3 ESRRRAs as the target in the Measurement List field. Next, press the Open button for folder and select the KEGG Legacy folder under the MSigDB folder. As expected, the results are similar to those from the Go term enrichment analysis we saw earlier. Now, let's set the mRNA targets gene set. This is expected to identify candidate mRNAs that may be involved in the downregulation of these genes. However, the reliability of this result may not be very high. Next, set the transcription factor targets. ESRRA targets are expected in this knockdown experiment, but many other transcription factors also appear. In the GSEA gene set, ESRRA is listed under the name ER1 rather than ESRRA. Also, it is not shown with the default filter settings, so you can see it by clearing all filters. Among the down-regulated genes, 8 genes were found that ERR1_Q2 predicts as targets based on sequence motifs. Press the preview button to look at these genes. Among these genes predicted to be ESRRA targets, we save the genes that are actually down-regulated under the name ESRRA target. These may become important candidates for further analysis. When we select these genes and load the GRCH38 version of the genome data, we can see that these genes are scattered across the chromosomes. Let's also look at the expression patterns of other genes predicted as ESRRA targets by ERR1_Q2. We search for them using the Find Measurement lists. Overall, among the ERR1_Q2 gene set, more genes appear to be upregulated. A motif near the TSS does not necessarily cause downregulation.